Hi, everybody. This is Peter Cooper from the NCBI. Welcome to today's webinar. I'll go ahead and turn this over to Mike Fiolo. Mike, take it away.
Thanks, Peter. Thank you all for taking the time to join today. I know you are all very busy. Today's webinar is dbGaP submission improvements and GaPTools. The webinar agenda, first I'll give a little bit of a background of dbGaP for those of you who are new to dbGaP. Then I want to talk about some of the submission improvements, specifically the study data outline, study configuration, and sequence metadata upload that we've introduced in the last 18 months or so. And then talk about our software tool called GaPTools, some of the current features, and Dale will do a software demo. And then we can have a Q&A afterwards. So for those of you who are new to dbGaP, dbGaP is a permanent archive of genetic data produced in research studies. It provides versioned release of the data for other researchers to access. It has two modes. One is a public metadata mode and a controlled access mode for individual level controlled data. The anatomy of a dbGaP data set. First of all, our high-level data model object is called a dbGaP study. And this consists of study metadata, which has information about the study, and then public documents. And these are documents that that are used within the study, like questionnaires or research protocols. And then we have a controlled access mode for the data that involves phenotypic data, genotype data called genotypes, sequence data, medical images, and association analysis. And all of these controlled access items also have metadata information that can be public as well. A research study is data collected from the same funding or research focus or cohort. That's loosely defined. Today, we do want to talk about some of the improvements we've made to the data submission part of our process. As many of you may know, and for those who don't, there's a study registration step involved with dbGaP, and that's because we have the controlled access data. And this step largely involves the principal investigator and their team and the NIH genomic program administrators registering the study and configuring the certification for releasing the data. And then there's a part of the process where the data is actually submitted to dbGaP, and that's where my team gets involved. And today we're going to talk about some of the improvements we've made here. So one improvement that we've made, and this is to reduce the iterations that we have often when we have submissions. Although the study may be registered with a certain data configuration, we don't always know for a specific version what data will be coming in. So to avoid having to ask that question, we ask that the submitter fill out a study data outline. And this is the submitter's assertion of what data will be included in that version of the study. This informs specifically what the curator should expect so that we know that we have the data that you intend to submit. And it also allows the submitter to see how the study was registered, because they may not necessarily be involved in that step. And because it collects the information that are typically needed by the curators, it reduces the need to ask common questions and to follow up later through email. Next, I want to talk about the study configuration web form. This is how we collect our study metadata. And previously, for those of you who have submitted in the past, this was collected through a Word document that was uploaded to the website. We've now replaced that Word doc with a web-based rich text form that allows you to enter all the information that was previously entered in the Word document to the site directly. And this improvement actually provides a way for the study session to be assigned as soon as the submitter is engaged with dbGaP, and it also allows a public preview of the public page right after the form is completed. The third improvement that I'd like to talk about on the submission process is the sequence metadata upload. And previously, the sequence upload to dbGaP, because it's handled through another team called SRA at NCBI, this was often done through emails that were arranged by the curators at the time of submission. But now we've kind of started to incorporate the sequence metadata upload through an automated email that's sent to the submitter as soon as the samples are loaded, and as long as there's sequence data expected in the study data outline. The upload is just through our upload page. There's a new section called sequence metadata, and that's where the files are uploaded. And this file is, unlike some of the other files that are uploaded to dbGaP submission portal, is downloadable so that others in the research study can access it as well, see what samples are loaded within this sequence metadata. This is a first step to automating the sequence upload and processing in dbGaP. We weren't able to do it all in one step, but I wanted to let you know that that's where we're headed. Next, I'd like to focus more on, once we get the data files, what we do to process them. And that involves a tool called GaPTools. But before I do that, I do want to step back a little bit, because I know that there's some people on the call that may not have submitted to dbGaP yet, or are involved in the registration phase. And I just wanted to go over at a high level kind of the relational model that we have in dbGaP and the identifiers that are very important. So we have two core files that are necessary for submission to for most dbGaP studies. And this is the subject consent file, which lists, it's a master list of the subjects within the study, and the consent of that subject. And the consent is the orderable unit within dbGaP. And then there's a subject sample mapping file, which is the explicit list of samples from each subject. And so these IDs are very important in the dbGaP model. There's a set of phenotype files, any number of them, that can be submitted along with the core files. And this includes any number of measured traits, and it can be any number of files. And so in this way, along with the data set, with the data, there's a data dictionary associated to all our data sets that are submitted on the phenotype side that allow us to configure any number of traits for a person. Next we have what we call the pedigree file. The pedigree file is only necessary when there's related people within the study. This file represents the relationships of the subjects to each other. We also have a file called the sample attributes file, and this allows you to include any number of attributes for a sample. The relationship of these files are the subject phenotype and pedigree files are related back to the subject consent file through the subject ID. And the sample attributes file is related back to the subject sample mapping file through the sample ID. Likewise, the sample IDs are expected to be into the molecular data, both called genotypes, any kind of omics data, and sequence data. We also require that the sex of the individuals should match in all the files, and that includes the phenotype and the genotype files. Now I'd like to talk about GaPTools and what it is. It's a validation software that we've developed to operate on the dbGaP formatted files. The current input is the subject consent, subject sample mapping file, pedigree file, subject phenotypes, sample attributes, called genotypes in both PLINK and VCF formats. This tool operates on all or a subset of these files. And the tool outputs QC results, some metrics and logging, and some reports. And if you have questions about specifics on this, the files within dbGaP, you can go to our submission guide, and the link is here. More about dbGaP tools. The current output returns specific errors and warnings of the validation, and some of the categories of errors and warnings are file format, data set to data dictionary matching, subject and sample ID matching across files, and variable naming, and the genosex and relationships to the phenotypic data. And then we have we have two reports that are reported out through the pipeline when appropriate, the genetic relationship and the X chromosome sex determination as computed by the tool. So you know, there is two validation modes for dbGaP tools. One is we run this tool internally on the files that you submit to dbGaP through our submission portal. It's as of this year, it's fully integrated with the submission portal, so that when you upload files, it will run on all eligible dbGaP submissions. When the tool is run, an automated email of the results is then sent to the submitter. And this tool also, within the pipeline, internal pipeline, compares the previously submitted files if they haven't been deleted from the submission. This allows us to do continuity across versions. But this tool also can be run in a standalone mode. It's set up as a Docker container, which is cloud ready. It validates files prior to submission. And this is the purpose of this is to reduce iterations with dbGaP. You don't have to submit the files. You can check them yourself if you're so inclined. It is a little bit technical, but it does run the same initial QC checks as dbGaP. Now, we do have a manual curation pipeline that is more expansive than this tool, but we are building out the tool to include what we can automatically. Now I'd like to transition over and allow software engineer Dale Conklin to show a demo of GaPTools.
Hello, this is Dale Conklin with the National Center for Biotechnology Information, which is part of the National Library of Medicine at NIH. I'm on the software development team and have developed with my team a tool that essentially takes the validation pipeline for dbGaP submissions and makes it externally available for anybody to download. The place to get started with this is to go to github.com to the NCBI organization and the GaPTools repo. So the URL you would go to is right here. And the first thing you take a look at is the README file. The README file talks about how to get GaPTools running on your local environment. The first thing you'll need is Docker installed. The next thing you'll need is to have Docker Compose installed. And you'll also need access to your data files. If they're local on your host that you're going to be running Docker on, you're fine. If they're located on S3 or GCS on the Google Cloud Platform, then you'll need to essentially make those files locally available to your Docker host. So we've included some links here for you to be able to go ahead and do that. And the final prerequisite is to make sure nothing on your computer is running on port 8080. So to do that, you take this command, run it on your host, and just make sure nothing returns. In this case, these connections are in time wait, and they're not actually listening. If this were listening, then that would be a problem. But since these are in time wait, they'll go away soon and won't be a problem for us. The next thing I want to go over is, before I go on to the setup, is actually the other README file. We have two markdown files in here. The other one is called GaPTools.md. And then this is a little more extensive README file. Goes over what GaPTools is. It talks about what is been updated in the most recent version, why we're using Docker to do this, the current quality control checks at a very high level, and then the metadata file. This is an important piece of the puzzle. You need to specify a metadata file and the called metadata.json, and its contents need to be structured like this. And this essentially tells the validation pipeline where your files are and what the file names are. Additionally, this talks about how you check processing status. It shows the validation pipeline, talks about all the checks in the validation pipeline, and at a high level what the checks do. And it talks about the output files and the results from the validation pipeline. And then we've included some sample studies, one without validation errors and a few with validation errors. So we talk about, and those are all based on the 1,000 Genomes project. So there's also a link to what the 1,000 Genomes project is right there. So going back to the previous README file, and remember, we've gone through our prerequisites, so we're good there. And then we go to our setup. So the first thing we do is clone the repository from GitHub. Let's go ahead and do that. And then we cd to the directory that just got created when we cloned the repository. The next step is we're going to make an output directory that we're going to use to output the results from the validation pipeline from our sample study. And then we're going to change the permissions on that output directory to make sure Docker doesn't have a problem writing to that output directory. That's it. Now we're all set up. We're ready to run the validation pipeline. So what we've included in this repo is a bash script, which essentially uses Docker Compose to start all the necessary containers that run the infrastructure for the validation pipeline. There's some important switches here you need to know. There's a -i switch. This is the input directory where your files are located. The -o switch, this is the output directory where you want the validation results to go into. And the -m switch essentially is the metadata.json file that I talked about earlier, which says where are your specific files, what are their file names of the files that are located in your input directory. So I'm going to go ahead and copy this and paste it in here and start that script running. So again, it's a Docker Compose script. What it does is it pulls down the NCBI/GaPTools container, which has our validation pipeline in it. It also pulls down several other containers to build the infrastructure to run the validation pipeline. We use Airflow to run the validation pipeline. So essentially what this is doing is building an Airflow infrastructure on your Docker host. So it pulls down a database container, a key value store container, a Flower container, so you can view your jobs, the Airflow web container, which is the user interface into the Airflow infrastructure, a scheduler, Airflow scheduler, the GaPTools UI, which is the UI into the GaPTools validation results, and then the Airflow worker container. So once all those containers are pulled down, then it begins to start the containers. It starts the database and all the associated components around Airflow. It then installs the validation pipeline into Airflow as what Airflow calls a DAG, a distributed acyclic graph. And then it initializes the database, it initializes Airflow, it brings all the containers up, and it makes sure that everything is running correctly. So it checks every so often to make sure until the Airflow web server is running, which is the final piece. So it knows once the Airflow web server is running, then the infrastructure is good to go. And once the infrastructure is good to go, then it kicks off the validation pipeline with the parameters you gave it earlier. Once this is done, it gives you where you can go to visit the GaPTools UI, and it also gives a command for what you do when you're done with the containers, how to bring all those containers down. So I'm going to go ahead and go into the GaPTools UI. There's an Airflow web console link here, which takes you to the Airflow web console. And this is the validation pipeline. All these boxes are different tasks that are run to validate the files that are part of your submission. You can look at the status of each of the tasks by looking at the color of the line that's surrounding each task. If you hover over here under the success, you can see which tasks have been completed successfully. The ones that are highlighted in pink are ones that have been skipped. This particular study does not have a pedigree file, so all the pedigree checks are skipped. And then the light green ones is the task that's currently running. And then the ones that don't have a status there's no border around it or a white border around it. So I'm going to turn auto refresh back on, and we can see that the validation pipeline has completed. So once the validation pipeline completes, now we know that our validation results are ready. So we go back to our main URL, and under here, we see the validation results are right here. There's a link that you can click, and it clicks to the validation results. We can see in this particular study, the files processed have passed. It gives you a link if you have any need for any help. You can click that and email the dbGaP SP help desk with any questions you have. You can see here there were 0 files with errors and 3 files with warnings. The warnings, this is a small study, so essentially the warnings are saying there were insufficient X markers to determine the subject's sex. And then there's also a validation file that is produced called combined one graph relationship. And I'll show you how to get to that in just a minute. There's also a helpful link here that you can go to, which shows you all the validation errors that could possibly occur, and then a description and the rule associated with that error. So going back to this email, these are the files that were analyzed in the report, and these are the files that you've specified in your metadata.json file. And then if you want to go back to the home page, you can go back to the home page. So again, in this email, there is this one file that's produced that you can take a look at. You have to go into the command line of your Docker host, go to the output directory that you specified in your output command line switch. And then in here, you go to the Geno directory, and then you go to the graph relationship directory. And then you can see the file that was produced as a result of this pipeline. And you can take a look at that file if you like. So that is the GaPTools tool as it is today. Again, a good place to get started with this is at GitHub/NCBI/GaPTools. And oh, one other thing I wanted to show you was how to bring the environment down. Go back to your GaPTools root directory of your GitHub repo and run the dbGaP-docker.bash command with the down argument afterwards. And that brings the environment down so you don't have containers running on your Docker host that you don't need anymore. So again, go to github.com/ncbi/GaPTools to get started with this publicly available tool. And I appreciate you taking the time to watch this demonstration, and have a good day.
Thanks, Dale. That was great. I think now we're at the point in the webinar where we can take any and answer any questions.
Yeah, Mike, do you want to show that slide, the one at the end with the just the help desk stuff? So yeah, Mike, we have some questions. One of them was, can you explain what the subject consent file is?
Yes, the subject consent file is, in its very simple form, it's a 3-column table. The first column is the subject ID of each study participant. The consent for that subject is the second column. And then the third column is the sex of the individual.
Okay, that sounds good. Thank you. A specific question: can the subject ID be the same as the sample ID?
Yes, that is allowed in dbGaP. We allow the subject and sample ID to be the same, although we treat those as 2 separate numbering spaces.
Okay, thanks, Mike. Another question: is dbGaP still accepting large batches of exome and genome data, or just genotypes at this point?
Right now, the GaPTools operates on called genotypes from either exome or whole genome. But in the future, we plan to expand it out. There's no plans to extend this to the actual sequence files, like a BAM file or anything like that right now. The SRA team has some tools to do that, so we're not planning to do that right now.
And somebody asked a question about the GaPTools in Docker. Dale answered it, but I'll just restate it. That you don't need to have the sequence files, that this is just to validate the metadata. That's correct, Mike?
Yeah, that's right. The tool will operate on any subset of the files. It does require the subject consent file, though. That is a required file. But any other files, if you want to just check the subject consent to the phenotypes, you can do that. If you want to check the SSM to the subject consent file, you can do that.
You know, one of the questions is, you know, do you absolutely need GaPTools? And the answer to that, I think, is no. You don't have to use it. It's just to help you make sure that your submission goes more smoothly, correct?
Yeah, that's right. One of the reasons we wanted to present this as a webinar is just to make people aware of this. Some of the people on the webinar are NIH officials that are managing the registration and the submission of the data, and others are submitters. Because we run the tool internally, a lot of users don't need to know anything about running the Docker version, but there are a fair number of submitters that submit frequently or have multiple submissions that might want to run this tool on their own. The other use could be a parallel resource that are managing similar data that might want to also run these type of checks. It does require the dbGaP formatted files, which is a relational model, so hopefully that wouldn't be too burdensome.
Yeah, so I mean, a couple people have commented on that, so it should help with your submission process. Back to the question that was about the submissions of the exome sequences. I think you answered the question about GaPTools not being a part of the submission of those kinds of data, just genotypes. I guess the question is, are you still accepting WGS data? Is there a capacity issue in accepting these kinds of data?
No, actually, that the capacity issue has been largely mitigated, namely because a lot of the data in dbGaP or available through access in dbGaP is actually hosted now on the cloud by some federated storage solutions that the NIH has developed. And so submissions directly to the SRA, if they're massive, petabyte scale, we definitely want to know about those before you submit them. But a lot of the issues that existed in the past about the storage space at NCBI have been mitigated. Hopefully that answers the question.
One question that came up was someone was asking what kind of computer GaPTools can run on. And Dale, I think we both answered that question in two different ways. So you were running that on the cloud, correct?
Yeah, it was a Ubuntu server on the cloud.
On the cloud, so.
Yeah, I was just getting a little more specific for that question.
Is it available and does it run on other platforms besides Linux?
Yes. So essentially anything that runs Docker will be able to run this tool.
So related to Docker, somebody asked about any experience with a Singularity container created from Docker. Do we have any experience with that?
Well, we looked into that, but we have not tested that. And as in its current form, we don't think it will work. But that is something that we may dig into further in the future.
Yeah, and if you're interested in this Singularity version of this, I think we need to hear from you. If we get enough requests, we might bump this up in priority.
So here's a question that might be a way to sort of summarize how GaPTools might help. The person says that they're going to be submitting data for a GWAS study in the next year or so and have only one previous dbGaP submission, but have not used GaPTools. What's the advantage for me to learn to use GaPTools versus submitting without it?
Well, I think, like I said, if you have one or two submissions, it's probably not something that you want to run as the standalone version, but knowing that it exists and that there'll be an automated processing of the data once it's submitted is hopefully helpful.
Okay, Mike, I think I want to wrap this up right now. And what I think we'll do is there are lots of questions in the questions pod that are highly specific and detailed. We're going to get these into a document. Materials in that document, we'll put that up there as soon as we have it ready. So we'll have all these questions answered for everybody.
Okay, that sounds great. And I do want to thank everybody for taking time out of their day to join today. Thank you.
All right. I'll go ahead and end the webinar for everybody.